Mendel's experiments on garden pea plants. See, Mendel found out 7 pairs of contrasting characters in pea plant. Such 7 pairs of contrasting characters controlled by 7 pairs of genes, commonly called alleles. 7 pairs of alleles. See, height of the plant, tall, dwarf. Color of the cotyledons, yellow cotyledon, green cotyledon. Then seed shape, round seeds dominant over wrinkled seeds. Then flower position, axial flower dominant over terminal flower. Then seed form in the sense round seeds dominant over wrinkled seeds. Then pod color, green color dominant over yellow color. Then next one about pod shape, inflated, constricted. So like that, these are 7 pairs of contrasting characters found out by Mendel in garden pea plant. Now, how he conducted experiments on garden pea plant? Before you go into that, Mendel found out there's something within the plant responsible for the formation of character. He called that something as factor or units, so which are now called genes. So actually Mendel proposed particulate theory of inheritance. Which is theory? Particulate theory of inheritance in the sense some particle within the organism responds for the production of character, responds for the formation of character. He called that something as a factor. The factors are called later on called alleles. Alleles are now called genes. So that's what given here. Mendel's theory of inheritance, known as particulate theory, establishes the existence of minute particles or hereditary units, factors which are now called genes. Actually, he performed artificial cross-pollination. Artificial cross-pollination experiments between two pure breeding varieties. So what is a pure breeding variety? A pure breeding variety is one which can able to give rise to same kind of offspring for many number of generations on self-pollination and self-fertilization. For example, you just see that tall cross with the tall, capital T, capital T, capital T, capital T, parents. In the sense, in the F1 generation you get, now you cannot call it as F1 generation, you get one, in the next generation you get tall only. So when it was self-pollination, this was selfing, you get only tall in the next generation. So this is what called pure breed. A pure breed is one which can able to give rise to same kind of offspring for many number of generations on selfing, self-pollination. Self-pollination means self-pollination and self-fertilization. So this is for pure tall. Then dwarf. So pure breed, dwarf into dwarf. Small t, small t, small t, small t. You get only dwarf. Again selfing. You get only dwarf. So like that, a pure breed is one which can able to give rise to same kind of offspring for many number of generations on self-pollination and self-fertilization. So he just always selected, Mendel's actually a pure breeding variety is one which can produce same kind of offspring for many generations on self-pollination and self-fertilization. So pure line breeds refers to homozygosity only. Homozygosity means what? An individual with a similar kind of genes, similar kind of alleles. Like that here, similar kind of alleles. So that's what called homozygosity. But actually pea plant is a self-pollinating one. Pea plant is a self-pollinating one. Why self-pollinating one? Pollen from one flower get deposited onto the stigma of same flower. Stigma of another flower of the same plant. So this is what called self-pollination and self-fertilization. Okay. So in that way, actually, pea plant is a self-pollinating one. Pea plant is a self-pollinating one. Why it's a self-pollinating one? Flowers are bisexual. That's why self-pollination is carried out here. But he made artificial cross-pollination. He made artificial cross-pollination by the process of removal of stamens. Process of removal of stamens or anthers. The process of removal of stamens from bisexual flower is called emasculation. Very, very important. The process of removal of stamens from a bisexual flower. So converting that plant as a female one, female parent. Right? Because if stamens are removed, self-pollination is prevented. So that's why he made artificial cross-pollination. The process of removal of stamen or anthers from a parent plant, it is called emasculation, but by emasculation, the particular plant is converted into female plant or female parent. So now, after emasculation, what happens? He collected pollen grains from another plant, considered as a male parent. Of course, that flower will have both. He collected only pollen grains from that, and then dusted pollen grains onto the stigma of female parent. So this is what artificial cross pollination made by Mendel. So after cross pollination, he collected the seeds. Pollination, fertilization, then seeds are produced. He collected the seeds and sown in the field to raise F1 generation. Of course, all the individuals produced in the F1 generation are commonly called hybrids. Then next one, he self-pollinated the F1 hybrids. Self-pollinated the F1 hybrids and offspring produced in the F2 generation. Yeah, and then next one about F2 generation, like that, F2 generation, F3, F4, like that. In each and every generation, he calculated the offsprings with the tall character, offsprings with the small character, probabilities. Then from which he could able to produce, he could able to get the results. Results means the ratios of monohybrid cross and dihybrid cross, like that. So this is what Mendel's experiments. Okay, Mendel, after collecting all that, Mendel died in the year 1882. Then during that period, none of them got interested on Mendel's experiments. They found they just not accepted Mendel's views. Only in the year 1900, Mendel's experiments were rediscovered and confirmed by three different scientists: Hugo de Vries of Holland, von Schermach of Austria, and Karl Correns of Germany. They just rediscovered and confirmed Mendel findings. So that's what commonly called Mendelism. So before you go into that, first one you must know about the terminology of allele. What are alleles? See, alternative form of same gene responsible for the determination of contrasting characters. A pair of allele, a pair of contrasting characters, or a pair of contrasting alternative forms of same gene responsible for the determination of contrasting characters. You just see that here, height gene. Height gene. It occurs in two forms. Which are the forms? Capital T, small t. That's what commonly called capital T into small t. Capital T into small t. This is what allele. So alternative forms of same gene responsible for the determination of contrasting characters. Tall, dwarf. Contrasting character. When you get tall character, you won't get dwarf character. When you get dwarf character, there won't be possibility of getting tall character. That's what contrasting character. Alternative forms of same gene responsible for the determination of contrasting character, commonly called alleles. Okay, what are alleles? The gene can exist in alternative forms. Alternative forms for the same trait. Same trait means character. It's called alleles. Then if an individual has two alleles are identical, if individual has two alleles are identical, such alleles are called dominant alleles. This one dominant alleles. If the two alleles are recessive alleles, homozygous. If the two alleles of in an individual, if the two alleles are same, called homozygous. And then homozygous dominant, homozygous recessive. If both alleles are dominant alleles, then called dominant alleles. If both alleles are recessive, called recessive alleles. Homozygous recessive, homozygous dominant, like that. Homozygous dominant and recessive, both alleles are dominant. Homozygous recessive, both alleles are recessive. Then these are always pure breeds. Homozygous individuals are always pure breed. Pure breed is called true breeding variety. Mendel always selected only pure breeding variety as parent. Pure breeding variety as parent. The next one, heterozygous. In an individual, if both alleles are dissimilar, in one dominant allele, one recessive allele, and this condition is called heterozygosity. Which are always heterozygous? Hybrids are always heterozygous. So this is what experiments conducted by Gregor Johann Mendel on garden pea plant. Then next one about molecular basis of flower color in pea plants.